Hello friends, in earlier lecture we discussed about the introductory part of our concept, that is germplasm conservation. In that we discussed the concept of germplasm, why the germplasm conservation is necessary, and we have seen the two different types of germplasm conservation. We just enlisted the names, we haven't gone through the details. In this lecture we will study the types of germplasm conservations in detail. So let us study, or let us go to learn the types of germplasm conservation. So we discussed earlier that these are in situ type of conservation and ex situ type of conservation. So we will see step by step. First we will see what is in situ conservation method. So let me take the highlighter. So what is in situ conservation method or in situ conservation? If we see the definition of in situ conservation method, it can be defined as on-site conservation of germplasm. Do remember, on-site conservation of germplasm is called as in situ conservation method. Or it can also be defined like in another way or another fashion, like preservation of the germplasm in their natural habitat. See, basically if we see the different biodiversity which occurs on this Mother Earth, it is distributed in very large area. So the area comprises different types of forest. Big forests are there, oceans are there, which holds tremendous biodiversity of the world, or the forest, as I told you earlier, forest, national parks, the sanctuaries. Okay, so all these things are now protected for, or they serve to conserve the biodiversity. If I ask you the different Biosphere Reserve, like Panchmari is there, okay, Nilgiri Biosphere Reserve is there, then different type of national parks are there, okay, for example Jim Corbett National Park, Kanha National Park, okay. So then your wildlife sanctuaries, Periyar Sanctuary is there, then Karnala Bird Sanctuary is there, okay, Bharatpur Sanctuary is there. So all these places are protected. All these places are protected so as to conserve the biodiversity. So what we do in situ. In situ, it literally means in their natural habitat, where they actually belong. For example, tiger resides in the forest. We have to protect the tiger in the forest itself. We don't have to bring it to some other place, some another place, and where we'll keep it in the protected environment. No. Or a deep forest where huge plants are there, or even seedlings or small plants are also there. So that plants are protected at their places only. Okay, where they are actually present, to which area they belong. If we consider different flora, that is the plants, or fauna like animals, different animals, tiger, elephant, all these organisms are preserved they are protected, not preserve is not a proper word. All these organisms are protected in their natural habitat where they actually belongs. Okay, so such type of conservation method is called as in situ conservation method. Okay, so that is what given or written here. Germplasms are conserved and protected in their natural environment where they naturally belong. They are protected there only. So if we see the example of in situ conservation, we'll have first one is Biosphere Reserve, second is National Park, third one is Wildlife Sanctuaries. So these three are the protected areas where the organisms, it may be flora or fauna, means plants or animals, they are protected there in their natural habitat. So there is a strict laws of the forest department which are to be implemented there, so that such as no any human interference or human activities are allowed in the core zone. They have separated some zone. They have also separated according to different zones, like a core zone or a buffer zone or a manipulation zone, like that, where percentage of human activities allowed, somewhere it is strictly prohibited. So in that fashion, all these laws are framed, all these protected areas are framed so as to preserve the biodiversity. Okay, so in Biosphere Reserve you will have you might know the Nilgiri Biosphere Reserve, then Panchmadi Biosphere Reserve in the MP, it is in the south region, Nilgiri, then Jim Corbett National Park, Himachal Pradesh, Uttarakhand, maybe that at that place. Kanha National Park is there, and wildlife sanctuaries, Karnala Bird Sanctuary is there. So these are the ways by which biodiversity, means germplasm, because their biodiversity is nothing but it is a germplasm, it is a source of live genes. Okay, live source of a gene. Okay, so these are protected at their natural environment. So that is about in situ conservation method. Okay, so you can see here Jim Corbett National Park. I have taken the images from internet, for which I have given the references. Like Jim Corbett National Park, you can see the tiger, the deer in Nilgiri Biosphere Reserve, or different birds in Bharatpur Bird Sanctuary. So here my impression to give this or my what we can say intention to give you these images is to just highlight the different types of protected areas, like national parks, like Biosphere Reserve, and like a bird sanctuary. Okay. Then second is Xc2 Conservation Method. So Xc2 Conservation Method means what? It is the off-site conservation method. See, opposite to the in situ conservation method. Off-site means what? Conservation, off-site conservation of germplasm is called as Xc2 Conservation Method, or conservation of the germplasm outside their natural habitat. This is also a definition. So in this case, what we do, we don't protect them at their natural habitat as we do in case of in situ conservation method. In ex situ conservation method, we bring it to some other place, or a safe place, or a protected place, and there we used to conserve that germplasm in the form of plant or in the form of animal or any creature. Okay, so if we see the example of it, different type of gene banks, different type of seed banks are there. Then botanical gardens, you develop in our college also. Botanical gardens is there, where the plants are brought from some other place. In our case, we brought it mostly from nursery or even from forest. If, for the purpose of conservation, we can brought the plants, or even in zoological park where zoo is there in Aurangabad. Siddharth Zoo is there. So there the animals are brought there and they are protected in that protected area like zoological park. So what we do in this case, we just bring a germplasm from their natural habitat and we protect in the other place or outside their natural habitat. Okay. In this method, genetic information of a germplasm is preserved in the form of in vitro culture. So I have written here, first thing is gene bank. Okay. So here some cells or tissue can also be preserved. In vitro means what? In vitreous, vitreous means glass. Okay. In vitro means what? In lab condition. Vitreous glasswares we use in the lab. So here we have to preserve the germplasm in a laboratory. Therefore, it is considered as a Xc2 conservation method outside the natural habitat. Then it can form a genetic information library which can be beneficial for many endangered, rare, wild and commercial places. Genetic resources are used for genetic improvement, biodiversity conservation, taxonomy, epidemiology. So these are some of the disciplines which uses the data of a gene or uses the genes for different aspects. So there are gene banks. If we go in detail regarding gene banks, which is a type of Xc2 conservation method, there are two different types of gene banks, in vivo and in vitro. As I told you earlier, the meaning of in vitro means in lab condition outside the natural habitat. Okay, and in vivo is opposite to that. In vivo means in their natural habitat. Okay, or in their natural conditions, rather to say natural condition rather than saying natural habitat. If you see the definition, banks in which genetic resources are preserved by conventional methods. See, we have to preserve seeds as it is or vegetative part as it is. Conventional seed banks, they are in vivo. We don't interfere with the seeds. We just only collect the seed and we preserve it. So it is an in vivo, in their natural condition. In vitro means a bank in which the genetic resources are preserved by non-conventional method. Non-conventional, such as tissue culture. So tissue culture is advanced method. It is a non-conventional method where we can preserve the different genes in the form of cells or tissues. Okay, so in the form of that we can preserve the genes or germplasm. So gene banks are having two different types: in vivo gene bank and in vitro gene bank. Both the banks are valuable sources for breeders to develop new and improved variety. Okay, so do you understood the Xc2 conservation method? It is the conservation of a germplasm outside their natural habitat, or it can also be defined as off-site conservation of germplasm. Okay, so these are the images which are taken from the source, internet source, for which the references are given. So these are the seed banks where this in vivo type of gene bank. In that seeds are preserved as it is, they are brought, and second is gene bank. So these are two different types of gene banks. So up till now we learned in situ and Xc2 conservation method. I hope you all have understood. In next lecture we will see the cryopreservation. Thank you very much.